All right, so let's wrap this chapter up. Still chapter 10, antimicrobial activity. Uh, Resistance. This is something that we talk about all the time in a microbiology class. You'll talk about it in your A&P class when you talk about treatment of disease. So, what is drug resistance? Drug resistance is simply an adaptive response uh, by which a microorganism begins to tolerate an amount of the drug that would normally be inhibitory. Uh, these can be due to genetic versatility and adaptability of microbial populations. They can be intrinsic, in other words, built in, or they can be acquired. Resistance to penicillin developed in some bacteria as early as 1940. So within just a few years, a handful of years of its development, it was already showing resistance. In the 1980s and 90s, scientists and physicians witnessed treatment failures on a large scale. Microbes become newly resistant to a drug after one of the following things occurs. Either a spontaneous mutation in critical chromosomal genes within the microbe, acquisition of entirely new genes or sets of genes via horizontal gene transfer from another species, or slowing or stopping of the metabolic rate so the microbe cannot be harmed by the antibiotic. These are referred to as persisters. Chromosomal drug resistance usually results from a spontaneous random mutation. You will see slight changes in drug sensitivity, but these can be overcome with larger doses of the same drug. So instead of giving, 25 milligrams 3 times a day for 5 days, you give 25 milligrams, or 40 milligrams 3 times a day for 10 days, or whatever. Horizontal transfer are the result of what are called resistance factors. These are plasmids found within one group of bacteria that contain an antibiotic-resistant gene. These can be transferred from one bacteria to another through conjugation. Conjugation would occur between bacteria of the same species, transformation, or transduction. And those last two can actually change these plasmids from one bacteria species to another. Transposons, also referred to as jumping genes, can duplicate and insert genes from drug-resistant individuals into plasmids. Those resistant genes can then be shared, and this accounts for the rapid proliferation of drug-resistant species. Natural selection can also play a role. Any large population of microbes will contain a few individual cells that are always drug resistant. If that population is exposed to the drug resistant population will have a selective advantage. Their offspring will inherit that drug resistance, and the replacement populations will evolve to have drug-resistant forms as the dominant species. So here is my original population. You'll notice that there's only 2 of these that have the resistance. We then give them the antibiotic. Most of these are going to be killed off, but these are not. So as those numbers rebound, you see a whole bunch of these and only one of these. And then what can happen is some bacteria will actually give their drug resistant to others, causing even more problems. So here's this one who is susceptible to this drug, but here through gene transfer, this gene is being transferred, and now this one and its offspring will develop the resistance as well. A threat report was issued by the CDC, Centers for Disease Control, Atlanta, Georgia, in 2013, which outlines a potentially catastrophic antibiotic resistant situation. That report said we may enter a post-antibiotic era where some infections will be untreatable. New and effective antibiotics have been slow to come to the market. Number one, because antibiotics are not economically lucrative. They take lots and lots of time and money to develop, and the return on that investment is generally not very good. Any of you who have gone out and bought an antibiotic and had to pay $75, $100 for it, or even more, would say, well, but remember, if you have a $100 prescription and they only get 1,000 of those filled a year, that's 1,000, two more, sir. That's $100,000. It may cost up into the tens of billions of dollars to develop these drugs. So, urgent needs. Clostridium difficile, C. diff, the number one cause of hospital-acquired infections. Carbapenem-resistant Enterobacter CRE, and drug-resistant Neisseria gonorrhoeae. So all of these organisms, Enterobacteriaceae is normally treated with this antibiotic here. However, there is a strain now that is resistant. C. diff is resistant to almost everything. Acinetobacter, resistant or multi-drug resistant, drug-resistant Campylobacter, fluconazole resistant Candida, which is a fungus. Lots of more serious threats. Some other concerns: vancomycin resistance, Staphylococcus aureus or bursa, erythromycin resistant strep, clindamycin resistant strep. All of these things are organisms that are now have strains resistant to the treatment of choice. Some new approaches to some antimicrobial therapies using RNA interference strategies, using small pieces of RNA that regulate the expression of genes, mimicking defense peptides. Again, peptides with 20 to 50 amino acids are secreted by mammals. If we can extract those and manipulate them, possibly we could use them as defense agents. CRISPR is a system found in bacteria that can cause very specific cuts in genes. We've mentioned CRISPR a couple of times already. This could very easily be used, or very possibly, I was going to say easily, very possibly be used to treat antibiotic-resistant infections together with an antibiotic. Virtually all microbes are non-cultivable, which means we can't grow them in the lab. So scientists are trying to figure out ways to grow and harvest their antimicrobial substances. All single-celled organisms, all bacteria, develop these antimicrobials because that's the only way they can survive, is by having a way to sort of keep themselves safe from others. So, but since most microbes we can't cultivate in the lab, we don't know what those different molecules may be. Bacteriophages, again, we talked about bacteriophages in the virus chapter. Bacteriophages are viruses that infect only bacteria. The former Soviet Union and other regions used mixtures of bacteriophages as medicine. Bacteriophage PA is used to treat ear infections caused by Pseudomonas aeruginosa biofilms. Other researchers are incorporating bacteriophages into wound dressings. The advantage of the bacteriophage is that they have very narrow specificity. They only infect one species of bacterium. Another advantage not listed, make sure it's not listed. Another advantage not listed is that they don't seem to develop resistance. The medications being used in the areas of the old Soviet Union, Russia, Ukraine, Uzbekistan, Kyrgyzstan, all of those places, uh, they're all using the same drugs that were developed in the 1950s and 60s. They haven't had to develop new ones because the old ones work continually. Probiotics, if you want to help nature, lots of folks now are consuming probiotics. You can buy yogurt with probiotics. You can buy probiotic pills and tablets. But these are live organisms fed to animals and humans to improve the intestinal biota. They can replace microbes lost during antimicrobial therapy. They augment the biota that's already there. They are safe and in some cases effective, and they are useful in the management of food allergies. Lots of folks with gluten sensitivities, for instance, are heavily into probiotics. Prebiotics are nutrients that encourage the growth of beneficial microbes in the intestine. Fructans encourage the growth of bifidobacterium in the large intestine and discourage the growth of potential pathogens. One of the newer things that have been developed is what is referred to as fecal transplants. And yes, that is exactly what it sounds like. It is being used to treat recurrent Clostridioides difficile, Clostridioides, commonly referred to as C. diff, and ulcerative colitis. You transfer the feces from a healthy person to the infected person via colonoscopy, and hopefully the colonies of bacteria on the fecal matter will help to displace the Clostridium difficile that has worked its way into the, uh, digestive system of the patient. There's work underway to develop a pill containing the species to recolonize the colon. I think I would much rather take a pill than have somebody else's feces placed in my rear end. But if I were to get C. diff and that was the only way to treat it, I would willingly accept the treatment. Drugs are administered to the host, even though the target is a microbe. The reactions between drugs and host. The effect of the drug on the host has to be considered. Again, early in this chapter, we talked about selective toxicity being ideal, but chemotherapy involves contact with foreign chemicals that can harm the host. Approximately 5% of all people taking an antimicrobial drug will experience an adverse side effect to that drug. Drugs can affect organs. They can affect the liver, the kidneys, the gastrointestinal tract, the cardiovascular system, and blood-forming tissues, nervous system, the respiratory tract, skin, bones, and teeth. There are certain antibiotics, if taken early in life, can cause really bad teeth reactions. So all of these things are toxic. Some major toxic adverse reactions: antibacterials, penicillin, rash, hives, watery eyes; carbenicillin, abnormal bleeding; ampicillin, diarrhea and enterocolitis. Actually, that ampicillin should be down here with another bullet point. Yeah, good. Cephalosporin can inhibit platelet function, decrease circulation of white blood cells, and nephritis. Formation of crystals in the kidneys can be caused by the sulfonamide. Those crystals can then block urine flow, and it can also cause hemolysis, a reduction in the number of red blood cells. Polymyxin can cause kidney damage, weakened muscular responses. Rifampin, damage to hepatic cells, dermatitis, all of these things. Some adverse toxic reactions to some other drugs. Antifungals disrupts kidney function. Amphotericin B. Flucytosine increased number of white blood cells. Metronidazole can cause nausea and vomiting. Some intestinal irritation, headache, and dizziness can be caused by some of the antihelminthics. Antivirals, acyclovir, can cause seizures, confusion, and rash. All of these things are adverse reactions. The allergic reactions that can happen to our antimicrobials. The drug actually acts as an antigen that stimulates an immune response or an allergic response. These can be provoked by intact molecules or by substances that develop from the body's alteration of the drug. Allergies have been reported for every major type of drug, but an allergy to penicillin is the most common. Sensitization occurs during the first contact with the drug. The second exposure can lead to hives, respiratory inflammation, or anaphylaxis. Biota. We mentioned earlier in one of the charts we showed the word biota, and I told you the biota were the normal microbes on a healthy body. They normally consist of harmless or beneficial bacteria. A few of them may be pathogenic. However, for them to be pathogenic, they have to move from one area of the body to the other. When it's on the area of the body where it's supposed to be, it is not pathogenic. If it moves to an area where it's not supposed to be, then it can become pathogenic. Broad-spectrum antimicrobials actually destroy the good biota along with the bad biota or the pathogens. Use of broad-spectrum antibiotics can lead to what is called a superinfection, where the microbes that were once small in number can begin to overgrow and cause disease. An example of a superinfection is a urinary tract infection. Lactobacillus in the female vagina are killed by the broad-spectrum cephalosporin used to treat the UTI. Then overgrowth of the Candida albicans occurs, causing a vaginal yeast infection or oral thrush. The fungus is always there, but under normal circumstances, the fungal numbers keep the bacteria number in check, and the bacteria numbers keep the fungal numbers in check. They keep themselves in balance. But when you take a broad-spectrum antibiotic like cephalosporin, that will kill the lactobacillus, and now the Candida albicans is free to grow and reproduce rapidly, and you get the yeast infection. Antibiotic-associated colitis, oral therapies with some antimicrobials has been associated with a serious, potentially fatal condition known as antibiotic-associated colitis or pseudomembranous colitis. Overgrowth of Clostridium difficile, Clostridioides difficile, invades the intestinal lining and releases toxins. It causes diarrhea, fever, and abdominal pain. This also, since it is inside the mucous membrane on the intestinal lining, is one of the reasons that C. diff is so very difficult to treat. For years, physicians have used what is referred to as a shotgun approach, using broad-spectrum antibiotic therapy for minor infections. This has led to superinfections and other adverse reactions. These adverse reactions can lead to the development of resistance in bystander microbes, the normal microbiota, that were exposed to the drug as well, leading to the spread of resistant pathogens. The growing awareness of this over the last 15 to 20 years has led to the reduction of this practice. It was for a long time believed that it was not harmful to just give multiple doses of antibiotics to people. We now know that absolutely is not the case. Tons of excess antimicrobial drugs in the United States are exported to countries where controls are not as strict. It is common for individuals in Latin America and Asia to self-medicate without understanding the correct medical indication. and drugs used this way are largely ineffectual and lead to drug resistance. That's going to wrap up Chapter 10. See you at a later date. All have a great day.